Hi everyone, greetings of the day. My name is Gaurav Singh and I am pursuing my Masters of Science in Agriculture at USQ. My topic of research is extent of carbon remobilization between tillers and mainstem in sorghum under a carbon stress condition. Let's start with the introduction. Sorghum is the world's fifth most important cereal in terms of both production and area planted. Sorghum belongs to the Poaceae family. Sorghum crop is well adapted to tropical conditions and it withstands relatively higher temperature during the growing period. It tolerates drought better compared to other cereals such as wheat or rice. As you can see from the graph, sorghum output in Australia was anticipated at 1.35 million metric ton in 2020 to 2021. The area was predicted to be 0.51 million hectares. Yields were projected at 2.65 tons per hectare, an increase of 0.65 ton per hectare or 33% over the last year. The significant rebound in production is due to much improved precipitation compared to the last season. Queensland is the top region by sorghum production in Australia. As an estimate, sorghum production of 2021 to 2022 in Queensland will be 1,734 metric tons, that accounts for 78.80% of Australia's sorghum production. What is carbon and carbon remobilization? Plant fix carbon primarily into 3-phosphoglycerate, which is a 3-carbon compound, hence the process is named as Calvin cycle. The other two variants of photosynthetic carbon assimilation are C4 photosynthesis and crassulacean acid metabolism. Plant use carbon dioxide during photosynthesis, the process whereby the plant converts the energy from the sun into chemical carbohydrate molecules. Plant use this carbon chemical to grow. Carbon accumulation and remobilization are critical mechanisms in plants for ensuring energy transfer between tissues with different functions or metabolic needs, as well as for the supporting new generation. Remobilization of storage material from vegetative organs to grain compensate economic yield, particularly in drought stress conditions. I have identified few major problems of sorghum cultivation in Australia. Sorghum is well known for its drought tolerance. However, one of the consequences of growing sorghum in a drought-prone region is stock lodging at the end of the season due to water stress during grain fill. Late season water stress on sorghum plants results in leaf senescence. This decreases photosynthesis, resulting in a loss of carbon production, which the developing grain requires. In response to this lack of carbon production, the sorghum plants robs carbon from the stalk and leaves to support carbon accumulation in the grain. This can result in lodging due to stalk collapse or breakage. Since the introduction of hybrid cultivars in the early 1960s, drought-induced lodging has remained one of the major issues for Australian sorghum producers. Despite the consistent prioritization of hybrid trait by plant breeders, drought stress in sorghum has been characterized at both pre-flowering and post-flowering stages, resulting in a drastic reduction in grain yield. In the case of post-flowering drought, lodging further aggravates the problem, resulting in total loss of crop yield in agriculture. The main aim of this experiment is to assess the extent of remobilization from unproductive tillers without panicle to fill main stem panicles under a carbon stress scenario. This research also focuses on whether remobilization from unproductive tillers could potentially reduce main stem lodging. Through this experiment, it will be evidenced that there is a vascular connection between main stem and tillers, which leads to the result that whether individual comes essentially act as independent unit or combined unit. For the background information, resistance to post-synthesis drought is important in Australia's northern grain belt since crops generally grow into water deficit. Symptoms of susceptibility to post-synthesis drought include premature leaf and stem senescence, lodging, and reduced seed size. Cheman et al. Reported a high frequency of water limitations in Australia's sorghum growing environments. A study conducted by Genome-Wide Association study detected 213 quantitative traits loci, the majority of which showed a significant association with leaf senescence. The high incidence of multi-trait association for the lodging quantitative trait loci indicates that lodging in grain sorghum is mainly associated with traits linked to carbohydrate remobilization. This research contradicts with the ideology of my research because in my research, leaf senescence is induced after flowering to remobilize carbon from tillers to main stem. But In the case of stay green research sorghum plant producing carbohydrate till maturity because of late season senescence. Positive correlation between plant height and grain yield have been reported for sorghum. The introduction of stay grain in sorghum and the associated reduction in lodging has opened the possibility to exploit this positive association. As you can see in the diagram for the control, I used it without any modification. In treatment T1, the plant was entirely defoliated and panicles were removed from the tillers to ensure complete carbon stress. And in the treatment T2, leaves were only removed from the main stem and panicles were removed from the tillers to induce partial carbon stress. Research design. I will complete my experiment in two different trials working with same ideology. I have already completed the first trial of my experiment in the field at Hermitage Research Station in Warwick. I performed my experiment with single genotype, that is Liberty, which included 8 replicates for the 2 treatments and 1 control. In this trial I used a split plot design for cultivation with the crop density of 90,000 plants per hectare on a 24 plots of 3-meter single rows surrounded by a guard rows. Topography of field contains black soil flat rising to moderately sloped cultivation, and this further rise to steeper cultivation and a grass country extending up to a high plateau. For my glasshouse trial, I will use control room for initial emergence of seedling in June because of unsuitable weather conditions, and then in onset of August I will place the pots in glasshouse for further growth and vigor. In this trial I will use randomized block design which consists of 8 replications with single genotypes of total 48 pots. I will cultivate my crop in pots of 18 liters in size, and the pot mixture will contain UK23 mixture of 80% potting mix and 20% coir mixed with fertilizer at the rate of dolomite 2 gram per liter, gypsum 2 gram per liter, osmocote 6 gram per liter and lime 2 gram per liter, and the mixture will be sterilized before sowing 2 seeds per pot. Suitable temperature required to grow sorghum in control room will be 30 degrees Celsius in day and 22 degrees Celsius in night. Duration of the experiment. The experiment was commenced in mid-December 2021 and anticipated to finish in mid of November 2022. Since beginning of my project, I have already completed my field trial, literature review and research proposal. Right now I am going to data analysis for my field trial experiment, and parallelly I am working on my glasshouse experiment. The glasshouse trial will be conducted from June to October, and for detailed timeline you can refer this figure, in which sowing of crop was done in last week of December and then I collected data on 17th February after synthesis. Later on I performed pruning on 23rd March and finally harvesting of my field was done on 12th April during maturity. Till now I have completed this portion of work and further work is identified as mentioned in the table. Data collection. In overall experiment, both field trial and glasshouse trial crop data are only used and collected at the time of synthesis and maturity. Morphological and biomass parameters, related traits linked to carbon remobilization will be considered between main stem and tillers. For example, stem length, stem diameter, leaf area, and leaf mass, etc. Traits linked to yield are only collected at the time of maturity, which are panicle numbers, panicle mass, and grain numbers per panicle. Statistical analysis. Standard analysis of variance will be performed between 8 replicates for 2 treatments and 1 control, and significant differences will be evaluated for dry biomass, leaf area, stem diameter, and other morphological traits. Replicate-wise comparison of mean data for various traits will be performed using the Fisher-LSD procedure. Correlation will be calculated between tiller stem mass and harvest index per replicate by using SPSS data software. Comparative analysis will be drawn between the effect of leaf removal on tiller stem mass for both treatment and control. Response curve will be generated for the effect of defoliation on growth of plant, photosynthetic activity and carbon production. Outcome of this experiment. Effect of both treatments will be significant for all the traits, and there will be considerable variation in extent of carbon remobilization, as a result of which potential of main stem lodging resistance will be varied among the treatments. When the coefficient of correlation is negative among leaf defoliation performed at the time of synthesis and the tiller stem mass, This indicates that there is presence of vascular connection between mainstem and tillers. When the coefficient of correlation is positive among leaf defoliation performed at the time of anthesis and tiller stem mass, then this indicates that there is absence of vascular connection between mainstem and tillers. Through this experiment, crop yield can be improved in drought stress condition. Crop loss due to stem lodging can be avoided via this approach. With the help of this approach, farmers can minimize climatic conditions for their crop, thus maximizing productivity, yield, and income. For concluding this presentation, I would like to address some key points again. Sorghum is an important crop for tropical climate of Australia, mainly grown for food and fodder. In Australia, sorghum cultivation have problems like drought-induced lodging. Remobilization of storage material in sorghum from vegetative organs to grain compensate economic yield, particularly in drought stress conditions. The objective of this research is to assess the extent of remobilization from unproductive tillers to fill main stem panicles under a carbon stress scenario. For achieving this objective, I have completed first experimental trial in field and I am going to conduct another trial in glass house. In this slide, I have mentioned all the references used and I am thankful to my supervisor for spending their invaluable time and efforts on me. And I have also so much gratitude for the examination faculty for helping me during the course of this research. I hope I provided satisfactory presentation.